This video is about the Cellular Respiration Lab, Part A. This setup, you'll realize, is a little bit old-fashioned, meaning it doesn't really use any nice equipment. We are very fortunate to have money in our budget to buy really nice probes and really nice electronic devices that can measure changes in gas levels, but a lot of schools don't have that equipment and don't have that financial capability. So why we look at this data and look at this lab is because College Board does believe that this is a quality, good setup for measuring respiration, and thus occasionally we will see questions on the AP test about this type of setup. So even if we were back in school in class, we would do this lab in the same fashion that we're doing now, just analyzing the data rather than actually doing it, because instead we would use a better setup that uses all of our nice equipment. In this lab, we are going to measure the amount of oxygen that's consumed by peas. So peas are a plant. Some of those peas will be germinating, meaning that they're growing. Some of them will be non-germinating, meaning they're still in like the seed form. Some of the peas will be kept at room temperature, so kind of cozy, and then some of the peas will be put into a colder environment. Our goal in the lab will be to analyze in which situation are the peas doing the most amount of respiration. We call that respiration rate. How quickly are they respiring? We will be using the amount of oxygen taken in to determine which peas are doing the most respiration. In this setup, we have a vial as pictured here on the lab. In that vial, the organism, meaning in our case the peas, are kept at the bottom. There is then a tube that comes into the vial, and in that tube there is a droplet of red water it's just water that has been dyed red. As the peas take in oxygen from their environment, that red droplet will be drawn toward the peas because the peas are like sucking in oxygen from the outside. However, also during respiration, carbon dioxide gas is produced. So since those peas are producing carbon dioxide, we would expect that red droplet then to be pushed back out. Well, if we want to measure the amount of O2 coming in, we can kind of negate or cancel out the movement caused by carbon dioxide. So we do that using a chemical called KOH, and KOH, potassium hydroxide, is put in the vial to absorb the CO2 so that the CO2 essentially becomes a solid and will not move that water droplet. So just to kind of recap here, so we've got our red droplet. As the pea is doing respiration, that red droplet will get sucked toward the peas, indicating that the pea is drawing in oxygen. Carbon dioxide is removed from the scenario by a chemical called KOH, and that KOH is going to convert the gas carbon dioxide into a solid so that the gas does not push the droplet back out. Therefore, any movement in the droplet will be caused by the sucking in of oxygen alone. The way that the experiment is set up is that it is kept in a water bath. In general, water just doesn't change temperature very easily compared to the air. So by putting these vials into water, it just helps to regulate the temperature. The long pipette is where the water droplet is, so that dyed red water droplet is put at the tip of each of the pipettes. Then, using a ruler, you can measure how much is that red droplet moving toward the organism in the vial. There is a control used in the experiment, which are glass beads. Beads made of glass are not alive. So I would expect to see in that vial no movement of the red droplet, because glass beads should not be breathing in or taking in any oxygen at all. However, when I look at the data table, I do see that there was some movement. So I'll conclude by quickly looking at this data table. This data table shows me that I have a vial of beads, and half of them are at room temp, half are at cold. My red droplet started on my ruler at a distance of 0.93. Over the course of 20 minutes, I saw that the red droplet moved to 0.90. So that is a movement distance of 0.03. Similarly, when I put my beads into the cold liquid, my red droplet started at 0.95 and it ended up moving to 0.93. Meaning that for my glass beads, my red droplet moved a distance of 0.02. That is not what I would expect. I would expect my red droplet to move no distance in something that is not alive. That means that there was maybe some changes in temperature causing a change in pressure in the vial, and therefore we subtract those numbers from the distance migrated for the peas in warm and the peas in cold. So to say that one more time, the distance for the bead should not have moved, but it appears that it did move. So I subtract that out from my other distances migrated to account for the fact that there may have been small changes in temperature or pressure in the container. Then, looking ahead, I can see that I tested my peas at room temp, my peas at cold temp. Those are both germinating, meaning they are growing. I also tested peas that were not growing at warm and peas that were not growing at cold. In this lab, we are focusing on the corrected difference, meaning the values that have been subtracted from the fluctuations in the beads alone. I'll talk through one of these. So for my dry peas, my water droplet started at 0.92. My dry non-germinating peas 20 minutes later had moved a distance of 0.07. Here it is, 0.07. However, my beads had migrated 0.03 in that situation, so I subtract my 0.07 minus my 0.03 to realize that my dry peas have moved a difference of 0.04. So I can then plot these on a graph to determine which situation caused the most movement in the red droplet, and thus in which situation was there the most respiration. So to wrap up here, you are going to produce a line graph that contains 4 lines on it. Each line will contain 4 points on it. So this will be a line for germinating peas at room temp. This will be a line for germinating peas at cold temp. This will be a line for non-germinating peas at room temp. And this will be a line for non-germinating peas at cold temp. So you are producing 4 separate lines. 1, 2, 3, 4. Each of those lines contains 4 points on it. You will connect those points perfectly, so don't do a line of best fit, just connect the dots, and then you will use that to answer the questions.